So welcome back, Ms. Cheryl Custard. Cheryl is a pharmaceutical consultant based in the US with over 24 years of industry experience in both pharmaceutical aseptic processing, training and qualification requirements. Cheryl also has a vast knowledge of small scale to large scale operations in both design, function, and qualification. Though much of Cheryl's career has been focused in microbiology and vaccines, she recently has expanded her talents to include immunotherapy. Cheryl is an active member of PDA and is a long-term member of PDA Technical Research Institute faculty. She holds a master's degree from East Stroudsburg University of Pennsylvania with a focus on biology and microbiology. Cheryl's area of expertise includes aseptic processing, training, qualifications, microbiology, vaccines, and immunotherapy. Let us welcome Cheryl with her presentation. Over to you, Cheryl.
Thank you, Subrata. It is a pleasure to be here with you guys this morning. I'm here in the United States of America, and I'm sure I'm having a little bit of difficulty hearing everybody right now, but as long as you can hear me, that is amazing. This is a little bit of a different kind of world for me because I am a very, very hands-on type of person. I don't like being across a computer screen. I would rather be with you folks in front of a classroom, or I would rather be doing some aseptic training live. I'm a hands-on trainer. So we're going to get through this presentation this morning, and I am so proud of my friend Subrata for initiating his company in the middle of a global pandemic, because that took a little bit of courage, in my opinion. But today's topic for the morning is going to be on, and I'm going to see if I can pull this up, because again, I'm having a little bit of technical difficulties this morning. I'm not sure why. Let me see here. Give me 1 second. I'm doing a screen share, but you can see this is not my strength. Okay, let me see if I can get this presentation to get pulled up here for you folks, because I had it here and it just sort of disappeared. But we're going to be talking a lot about microbial out of specifications. Subrata, can you let me know if you could see my presentation in front of you, or am I still having some share screen issues? Okay, good. As long as you guys can see it, I'm fine. So this morning, this is going to be an abbreviated version, I guess you could say, of handling out of specification results for microbial issues. And in my opinion, this could almost be a day-long event. There's a lot of information to cover in this particular topic. So I did my best to do an abbreviated or compressed version of this for you folks this morning, or should I say this afternoon. A little bit about myself, and I know Subrata did a very good job introducing me to you folks. Let me see if I could do, this is what I look like. I think I also see you guys on my screen share. And in case you want to have an idea of where I am in this great big planet right now, that little red dot on the United States is Pennsylvania. And I'm one state below New York City, just to give you an idea of where I am located. I have about 5 inches of snow on the ground. I know that my buddy Hal, I know you just saw his dynamic presentation. He is in sunny Florida, where they are having a cold spell themselves. But that's one of the nice things about doing online webinars and online events, is that no matter what the world looks like or how the weather looks like outside, we can still reach you guys and give you guys a dynamic presentation. So I'm not going to do any real poll questions for you this morning. What I would like to do is ask you to have some food for thought. And food for thought means really think about your company, think about yourself, and think about how you would answer these two questions as we go through this particular presentation. Let me see here. From current slide. Let's see if we could get that up. How important is data integrity in determining product quality at your site? And is data integrity important in determining a microbial out of specification. So again, how important is data integrity in determining product quality at your site? And is data integrity important in determining a microbial out of specification? The reason I bring up data integrity is data integrity almost drives everything that we do in the industry. And I'm going to get into data integrity very briefly. Again, that could be another day-long event. But data integrity means that we can actually prove that what we're putting out the door meets the GMP requirements for a safe product. That it's safe, it's pure, it's free from contaminants, it's made under GMP conditions. There's a lot of different qualities that go into whether or not we can actually sell a product on the market. And data integrity, the quality of the data that we put out there determines whether or not that's true or not. Now, when it comes to a microbial out of specification means that we had an issue. That during one of our testings, during one of our situations in a lab, we identified an issue. So we now need to use data to support whether or not that issue is going to impact whether or not we're going to be able to sell product, whether or not we're going to be able to move forward with product. So these are the kinds of things that I would like to focus on in this presentation, in addition to going over a high-level overview of how we're going to go about doing this. So again, very brief introduction, I should say, in data integrity. We're going to talk about the process, the rules, and expected outcomes for handling out-of-specification investigations. We're going to talk about the different phases of an OOS investigation. And then finally, throughout the presentation, we're going to cover the rules, regulations, and guidances governing an OOS. An OOS, in case you haven't figured that out, is out of specification. So a brief introduction, I should say, into data integrity. So data integrity by definition, and if you look it up in any regulatory document, you're going to have a pretty similar definition. Data integrity is the degree to which data is complete, consistent, accurate, trustworthy, and reliable. And so what are the different things that we need to use or look at when it comes to data integrity? What exactly is data integrity being used for? Is it being used for product release? Is it being used to move the product onto the next level of manufacturing? Are we using it to defend whether or not our product is sterile or not? And I know Hal did a fantastic presentation on sterility assurance. What exactly is this data being used for? What exactly is the data being used to defend? It also applies to retention. How long do we have to retain our information for? What is the expected expiration date of our product? How long can a product be considered or recallable in the event that it goes into a client? Retrieval. How fast can we pull the data out? If you use Part 11 compliance systems, for example, where we use learning management systems and software, if you're in the middle of a regulatory audit, what happens if you lose power and you can't use your computer systems? What happens if your computer network goes down? And you might say that'll never happen. Oh, I've been in big industry before, folks. This has happened where you go to pull up a document on your learning management system or your document management system, and you can't because your entire network just went down in the middle of an audit. Do you have a paper trail? Are papers even still being used at your site, or are you completely electronic? What are your fail safes? What are your backups? And also, how do you archive data? Once it's no longer needed, once that product is considered no longer needed on the market, destruction is a very harsh word, but realistically, do we have to keep things forever? What if we make individualized or customizable products and the patient is no longer alive? Do we have to still keep that information? Because it was made just for that client. And a lot of times when it comes to antibody therapy and immunotherapy, that is a very, very real thing where we make customizable products just for one particular client. And going along the same lines of data integrity, it would be very important to go over again the very basic concept of ALCOA. And if you've never heard of ALCOA, it's mostly a good documentation concept that is being highly recognized in the industry. Anybody that does good documentation training will show you a little bit about the ALCOA model. And ALCOA stands for accurate, legible, contemporaneous, original, and attributable. So by accurate, it means free from errors. And if errors were made, that they were corrected according to your GMP and also company guidelines, and that the documentation contained in that information is defendable, legible, permanently recorded with ink, usually if it's on a piece of paper. We can't use something that's erasable, and it has to be human readable. So if an auditor were to sit down and look at your documentation, they can actually see it. Contemporaneous. Dates are identified with the date, time, and that everything can be traceable. Original, true copy. It can't be a photocopy. It cannot be. Now, if you do scan documents into a data management system, that is a defendable case. But in many cases, the regulator still wants you to maintain or keep your hands on the original document. And finally, attributable. What does that mean? That means that the source of the data identified has the process and the person visible. I need to put this on full screen mode. I'm not sure how to do that. I'm trying, guys. Slideshow from current slide, and it's not there. I thought I was. Okay, maybe I'm not. All right, so here we go. We're going to get through this. As long as you guys can see my screen and you can hear me, we're going to get through this just fine. So what are some of the rules and regulations concerning laboratory records? Because we really need to start talking a little bit about how data integrity applies to our laboratory records, but also how data integrity applies to out-of-specification results. So under 21 CFR, and this is US FDA 21 CFR 211-194, and I'm not going to read the entire CFR to you, but I did take out some bullet points that I thought were relevant to this particular presentation. Laboratory records shall include complete data derived from all tests necessary to assure compliance with established specifications and standards. What do they expect from us? They expect a description of the sample received for testing with location where sample was obtained, how much of the sample was obtained, the lot number, the date the sample was taken, the date the sample was received by testing. So they want acknowledgement that the sample was received by the testing department, because when it comes to time-sensitive information, we need to make sure that it got to our quality control unit in a timely and effective manner. They want a statement of each method used in the testing of each sample. They want a statement of the weight or measure of sample used for each test, so how much of the actual sample was used for each one of these tests. A record of all calculations performed. Initials or signature of the person who did the test, so the person who did the test, the performer. The date the test was performed, and the initials or signature of a second person showing that the original record was reviewed for accuracy, completeness, and in compliance with established standards. Continuing with US 21 CFR 211-194, what do they also expect of us? They want complete records that shall be maintained of any modification of any established method. So if we have to make a change to anything, they want this to be the standard. Such record shall include the reason for the modification and data to verify that the modification produced results that were at least as accurate and reliable for the material being tested as the established method. Complete records shall be maintained for any testing and laboratory standards. Complete records shall be maintained of the periodic calibration of the laboratory instruments, such as our recording devices, our micropipetters. Any equipment has to have a calibration sticker saying that it was shown to be in a state of compliance when it was being used. And they also want complete records that shall be maintained of all stability testing. And there's another 21 CFR reference. So we're not going to go into every one of these 21 CFR references. What I would like to go into is more of our guidance documents and also a roadmap, a plan of how to actually conduct these out of specifications. And it wouldn't be a presentation if we didn't talk about our EU folks a little bit. There's still a lot of work going on when it comes to the EU drafts, the EU document revisions. But under UDRLEX Volume 4, Part 1, Chapter 6, I like to give you guys complete references, because if you ever want to look anything up, it's nice to know where to go for your sample. Under quality control, laboratory documentation should include a procedure for the investigation of out of specification and out of trend results. Trends should be evaluated and periodically reviewed. I like to call this thing predictive deviations. And I'm going to go into that as soon as I finish this slide. And any OOS, out of specification or out of tolerance, OOT, results to be subject to an investigation or an incident report, whatever it is you guys like to call it. So what do I like to call a predictive deviation? And I know you might say, well, if you could predict it, then it shouldn't be a deviation. A lot of times you can actually see seasonal variations, or even when you have to increase manufacturing, you're going to start to notice that when you have to increase your batch sizes, you're going to start to see an increase in environmental monitoring, for example. Or your samples may not be as clean as you'd like them to be. By the use of trends or predictive deviations, as I like to call them, you can actually build safeguards into your programs to counteract this. For example, if you know your environmental trends are going to increase every time the weather gets nice outside, and I know that in Pennsylvania, we have 4 seasons here. Every spring, as soon as things start to pollinate, as soon as everything comes to life, pollen shows up in the air. And when the pollen shows up in the air, it's a particle that we know is coming. It's a particle that, yes, it even causes allergies in us. Trust me, I have the worst allergies. So I can predict now that in the spring, I'm going to have allergies, just like I can predict now that in the spring, these particles can have a potential trend or a potential issue on my manufacturing suites. So we're going to change potentially our cleaning program to accommodate for this particular event that we know is going to happen every spring in the state of Pennsylvania. Now, I like to use that as an example because what happens in Pennsylvania may not happen where you are in the world. You may have other incidents or events that happen in your part of the world that you need to accommodate. But you can already predict that once that time of year, once that event happens, you're going to have an out of trend or an out of specification. So because of that, you actually could almost plan to how to counteract that issue or how to handle that situation before it becomes a problem. So I'm going to go a little bit now into the actual guts of investigating an OOS test result for pharmaceutical production. Again, this is a high-level overview. This could be a much longer course, but again, we only have a very limited time this morning to talk about this. So I want to give you some of the reference documents, as I already mentioned, but also what exactly is the expectation, the different phases that goes into these OOS or out-of-tolerance test results. So the first thing we're going to do, and this is again the basic 5-step model in my opinion of how we're going to perform this, we're going to have the potential OOS identified. Once we identify it, we have to do what is called a phase 1 lab investigation. I'm going to go into all these for you, so I'm not going to get into every one of these right here. Once we do our phase 1 lab investigation, depending on the outcome of that, we're going to move into a phase 2 lab investigation. We're going to talk about when is retesting or resampling appropriate, and we're also going to talk about reporting considerations. So we're going to talk about the potential OOS identified first. So once we have a potential OOS, and we don't like to automatically say we have an out of specification. We want to sit there and say we found a test result in our micro unit that was not according to our sample specifications. So we need to investigate what happened. Is it the sample? Was it a test method? But before I get ahead of myself, first of all, conduct a review for data accuracy before the tests are discarded. Make sure that the test was performed properly, that the technician was trained on the test. Make sure that there was nothing unusual about the lab that day that could have contributed to the OOS. Because an OOS means that the sample actually is compromised. If review of data could not identify any significant errors, you then have to initiate what we're going to call a full-scale OOS investigation. So if during your interview process, if during your initial review, you notice that the person used a wrong reagent, a wrong I'm trying to think of the word for it here, standard. That is still a bad thing. I'm not going to negate that. That is a human error related thing, and that needs to be addressed. But does that mean that my test was wrong? Does that mean that my product is compromised? No. It means that the operator performing the test made an error, the error was identified, and that when we performed the test according to our SOPs accurately, the sample actually came out fine. If that could not be identified, we then have to go into OOS investigation mode. Why is this sample not meeting our testing standards? Again, note, if the out of specification was identified by a contract lab, because sometimes we outsource our testing, the contract lab must notify the company of their OOS data and findings, but it's the manufacturer's job to initiate the OOS investigation. The contract lab only reports on their findings. They don't initiate the investigation. It's our job as the owner of the sample to do that on our end. As I mentioned, now that we're in full OOS lab investigation mode, we're going to go into what is considered, or what is called by the industry, a phase 1 lab investigation. During phase 1 lab investigation, you ensure the analyst followed procedures again. Is there anything, again, that the analyst did that could have contributed to the OOS? We're going to verify calculations. Now, if you're using calculators and not validated equipment, make sure that you're using more than one calculator. An example would be, in my previous years, I would always perform calculations using a calculator in my lab. And we would get a printout. You know, there would be a printout. The printout would be attached to my paper record. Again, I'm going back a few years, guys, but I would attach the printout to my paper record. My check signer, my second verifier, used the same calculator to verify calculations, and they also got a printout. So it showed that the printouts matched, okay? Came to find out that when the document was sent to the next level of verification by quality, quality performed the calculations on another calculator, and they got a different result. Came to find out that the calculator I was using was not exactly working anymore. So we really weren't checking the calculator to make sure 2 + 2 made 4. We just trusted that the calculator was giving us valid results. And my check signer used the same calculator, so there was really no way to prove that the calculator was breaking. It broke the same way twice, in a sense. So you want to try to make sure that if you are using calculations, that you use validated systems to do this, and that if you're using still very handheld or rudimentary systems for doing this, that you have separate calculators or second calculation devices to make sure that the results are accurate. You have to ensure equipment was performing properly, that it was validated and calibrated. Another thing I like to ask my operators or my analysts are, did you drop something? An example would be if they dropped a micropipetter. You don't want to assume that people know that dropping things can actually make things go out of calibration, especially if it's a new technician or a new operator, or because it's an expensive piece of equipment, they may not want to bring it to your attention. So make sure you ask them the right questions. Was it performing properly? Well, to the best of my knowledge, it was. I can't do a live check on my micropipetter, perhaps, to make sure it was performing properly. Did anything happen to the micropipetter before you used it? Did you notice that if anything sounded wrong with it? Did it seem to perform properly based on your normal performance of this equipment? Is it potential that it was dropped, as an example? Check your standards and reagents for expiration and the correct one was used. I like to say that this is a no-brainer, especially when it comes to electronic systems, because we can usually scan barcodes in, but it's amazing how many times that you are supposed to document things again concurrently before you use them. And people actually sometimes document things after they use them. And that's when you're going to find that perhaps the wrong equipment or the wrong expiration date was used, especially if you're still using paper modules. Check operator qualifications again to ensure they're up to date on all training and document revisions, because if a standard was changed and they did not train on that standard because they were not here, maybe they were on holiday for the last 2 weeks, they might have been actually doing the test based on an old standard. Yes, the SOP should be with them when they do this, That is good documentation. That's good manufacturing practices. The GMP say the document, the SOP should be with you when you do these tests. That doesn't mean that they're actually doing that. What people should be doing and what people actually are doing can be two different things. And I would be remiss to say that I actually have never had a human error on my end too. I'm human. So I have made mistakes in a lab, and I was not one to catch it. It was actually my management or my quality unit that caught those mistakes. So that's why we have all these checks and balances in place, because we're only human. We're going into a lab. We're going to do a job to the best of our knowledge. I don't think anybody's going to go into a lab and say, how am I going to make a mistake on this test today? But again, the human aspect of everything we still do is a huge contributor to how the outcome of all these tests and also the product disposition really is. Another side note I'd like to bring up, lab errors should be relatively rare. There shouldn't be a lot of lab errors. Otherwise, you have an issue again with data integrity, especially when it comes to sterility testing or product release testing. If clear evidence of error occurred, the results can be invalidated, but you always want to make sure you discuss this with your quality unit before you do this. If a lab error cannot be clearly or subjectively identified, we're now going to move into what is called a phase 2 lab investigation. So what happens in a phase 2 lab investigation? Another side note, identifying the root cause of the out of specification is the main goal of a phase 2 full-scale lab investigation. We're not going to guess. We have to come up with a definitive root cause for why this test result was out of spec. Otherwise, we may have a product that may not be able to be sold on the market. The investigation should include a well-documented review of production and sampling procedures and data. We're not just going to blame the QC analyst here. Never once do I like to say that the out of specification was a result of a poor laboratory test. The sample came from manufacturing. Did anybody interview the manufacturing staff to find out if there was anything that could have compromised that product, compromised that sample? Was the sample even taken properly on the manufacturing side of things? So we don't want to automatically assume that this is a laboratory error per se. We now need to move beyond the lab and start to question other people, other stakeholders, to find out if there was any issues during the entire, processing of this product. We need to have a clear statement of what is being investigated and if a trend is identified. The definition of a trend, I think sometimes it depends on whether or not, on what the trend actually is. Sometimes one time is too many. But if you're starting to see the same issue showing up again and again and again, and I know in the United States we have this saying that the definition of insanity is doing the same thing over and over and over again and expecting different results. So if you're seeing a trend forming, it means that there is something breaking or broken somewhere, and you need to find where the breaking is occurring and you need to correct it quickly. You've got to remember, all of this is transparent during a regulation, and I'm not scared of regulators. I'm not scared of regulators. You might say, oh, well, we just don't want the regulator to find it. Why not? See, we're so afraid of audits. The thing is, we should be more afraid of hurting clients. We shouldn't be afraid of the FDA or of the European Union. We shouldn't be afraid of anybody coming to our site and finding a problem. It's their job. It's their job to make sure that the product that we're putting out the door, they're our quality check. We would like to think we're doing a good job, but it's their job to make sure that the product we're putting out the door is safe. And if we're not doing that, then we should be fixing that. So never be afraid of a regulator. Never be afraid of an audit. Always embrace it, because they're your quality check. They're the ones who are making sure that you're actually still meeting quality standards of the product that is going out your door. Additional testing might be needed. There might be some retesting or resampling required. And of course, there has to be an impact statement on your released product if it's already released, or your supply chain. So why would a released product be impacted? For those of you who are not fully aware of how released product works, and I'm going to give you a high-level overview of how this works. When you release a product to the market, there is an industry standard, there's a regulatory standard that says that you still need to test that product while it's in the market to make sure that it still meets the same quality standards 6 months after you sold it to the market, 1 year after you sold it to the market. It's not you release it to the market and then you forget about it. You have to prove to the government that as long as that product is on the shelf and can be used by human beings, and as long as that product is in a human being, you have to prove that it's never going to harm a human being. And that's even after it's been sold. So what we're going to do is we're going to pull retention samples that we put into a unit, and we're going to test them to make sure that the product is still safe, pure, stable. We have to test it for stability. We have to make sure that we don't have to do a potential recall 1 year after we set it to the market because we're having issues with that product. What if during this retention, this retesting phase, in a sense, when we have the product on the market a year and we're pulling samples to show that it's still safe to use, that's when an out of specification shows up. Our stability sample showed that there was an issue with our stability. We now have to decide, was the test done improperly, or do we really have an unstable product on the market? And we need to make sure that when we're looking at the released product, do we have to do a recall at this point? Okay, so we need to make sure that what happens if there's no real clear root cause? That's a concernable. So if we cannot come up with a discernible root cause, again, that's why quality's job is sometimes one of the worst jobs in the world. I've been on both sides. I've been on the manufacturing and quality side of a lot of things. You now need to decide product risk. You have to almost do a quality risk assessment to determine whether or not the out of specification will have a recallable risk in the future. And a lot of that is also based off of industry trends and trending. Trending is huge. If in the past you've seen that if a sample goes out the door and maybe it's a relatively unstable product to begin with, but it's a necessary product, these are the kinds of things that you have to weigh in from a quality risk perspective. And so your quality risk management team, as well as your quality team in itself, there is actually quality risk guidance documents out there that map out how you should do a quality risk assessment. And I don't have the time to go into a lot of this for you guys today, but I just like to bring that up. There's so many different tools and resources that are available to you folks. You just have to navigate. And that's one thing I've learned how to do as a quality unit is I've learned how to navigate all of the industry regulatory websites. You always like somebody to do the work for you, but the reality is sometimes you just have to go on there and find it for yourselves because all this information is out there. You just have to dig deep into it. Being that I'm having issues with audio, I know Subrata is giving me some questions, so I'm gonna try to see if I can answer some of these now. What are the actions to be taken when sterility failure attributed to a lab error? Sterility failure attributed to lab error. So I'm assuming that question means if I made a mistake in the lab and it caused me to have a sterility failure, what do I do? Well, when we have a sterility failure, one of the first things I look at is, were additional product samples taken? Okay. Yes, sorry. We will come back to that question. So going back to our phase 2 lab investigation, involve all other departments that could be impacted. And I like to, and that also goes along the lines of sterility failures. And I apologize, I do need to have a drink once in a while. I just can't talk nonstop without having something. Involve all other departments that could be impacted. And I also like to include my manufacturing areas. Contract labs should be included in the investigation if you use them. And if an OOS investigation confirms the failure is a true OOS and root cause is identified, the investigation may be terminated and product rejected. We don't want to sell product that actually is bad, obviously. We're not trying to test into compliance. If we find that there is actually an out of specification, can we realistically sell product? Can we say that, we have standards, but if we don't meet all the standards, that's okay. Then what is the actual point of having release standards? So it goes back again to our data integrity discussion in the beginning. What exactly does this data mean? And if the data means something, you have to actually use that information to evaluate your quality call on whether or not the product is safe to sell or not. Again, nobody likes to reject products, so this is a very difficult decision to make. And sometimes you do need to contact, and I know people are afraid to do this too, you sometimes have to contact your regulators. You need to contact your regulatory units and say, I don't want to reject product, but this is the situation we're having. And you might say, well, Cheryl, I don't want to bring this to their attention. They are going to already know about it. Anytime there's a sterility failure, it's a recordable or reportable event. If you have a product failure, it's almost a reportable event. You can't hide it from the regulators. You do have to have these as reportable events. They're going to find out about it anyway. So being transparent and making sure that you're making the right quality call is very important again. Again, note, if the investigation should extend to other batches or products, or I should say the investigation should extend to other batches or products if deemed necessary through trending or interviews. So if for some reason we found that one of our technicians was consistently performing a test incorrectly, just because other test results pass does not mean that the other products are safe. They might actually have a stability issue a year from now, as an example, because they might have performed the test, it gave us a result. That result we were hoping through a data integrity standard was a stable test result. We found out that the technician didn't perform the test properly, was consistently not doing it properly, and now the test results, we now have questions whether or not the test results we received were good for making a product disposition statement, a product release statement. So what we're going to do now is possibly pull some of our retention samples, retest them to make sure that they're still meeting the same quality standards that we expected when we first started this whole thing. When is retesting appropriate? Again, this is all based off of guidance information. Again, every product you make is specific to your company. You know the stability of your product, you understand the components, hopefully, and the composition of your product and processes. So all of these little factors have to go into whether or not you decide whether or not to retest, resample, to sell or not to sell. No one's going to be able to make that decision for you but you and your quality unit based off of your quality risk assessment. So when is retesting appropriate? According to some of the standards out there, when the number of samples to be tested must be specified. When the number of samples to be tested must be specified, do not test into compliance. So an example would be here. If I have to stability testing on 6 samples, right? But I say to myself, well, you know what? If I do 8 samples, and I think I might be getting ahead of myself here, and if I average those 8 samples, I might actually get a passing test result. So averaging is only good, and I think I'm going to be getting ahead of myself here when it's appropriate or identified in the test. And also averaging is only good when you use the right number of samples. So you can't just say, I'm going to test 20 samples today until I get one that passes. You just can't do that. You still have to follow your procedures. You still have to follow the GMPs with the test when it comes to that specified test. And in some cases that is specified in the regulations. That is not something that is for you to decide. If possible, test the original sample that led to the original OOS. Excuse me. This is also useful. Retesting is also useful when you might want to question your equipment. But now you have to say to yourself, if I'm questioning my equipment and my equipment was calibrated 3 months ago, if I find an issue with this equipment, anything that I tested for the last 3 months is now in question. Tests should be performed by a different analyst from the first, but with at least the same experience and knowledge as the first. So I'm not going to have a 5-year experienced analyst. The OOS was performed and was identified by a 5-year experienced analyst. I'm not going to have the retest done by someone who was signed off a month ago. I need to have someone who's been on the site a couple of years, perhaps, with the same level of knowledge and competency as the first analyst. If a retest clearly indicates lab error, substitute retest samples for the original. You can actually, if it's possible, say it's a final product, and before the product goes to market, we can actually pull additional product samples for our tests. And if no clear indication of error, this must be considered again when it comes to your batch release process. When is resampling appropriate? Now, this was not very clear in a lot of the regulations or guidances. There was a very, very small amount of information you could find on this. Resampling is appropriate. What we could do is we can collect additional or new samples for a batch if a sampling error was identified. So if on the manufacturing side of things, a sampling error was identified, or perhaps we should have pulled 10 samples, but we only pulled 5, for example. For our initial testing, we can collect additional or new samples. But again, there's very, very little guidance when it comes to resampling procedures. Also, reporting considerations. What does that mean? We talked a little bit about averaging on the previous slide. Averaging should not be used for unit results, and this actually comes right out of guidances, not to be used for unit results, tests for variability, or to combine with the original and retest results, because this can conceal variations. If you have, example, 5 samples, and 5 samples were meeting criteria and you have 1 outlier should still be investigated. Why? Because if you average those 5 samples, you're still going to have a passing test result. But that 1 outlier is still an issue because you shouldn't have an outlier. So why were 4 good and 1 bad? What's up with the 1 bad sample? So that still has to be considered even if averaging is part of your original criteria. You should only be used if averaging is part, again, of your quality procedures and used during the original testing. And identifying outliers may be an indicator of an unstable or a non-uniform product. Again, another side note, not appropriate for use with tests that check for variability, as already mentioned. So how do we complete the OOS investigation? And again, all of this is based off of some guidance information and also industry trends and standards that are out there right now. And this is pretty much like a global concept. It's from the MHRA, it's from ICH documents. Invalid results where a root cause is found do not use to evaluate batch quality. Confirmed test results include in batch quality and batch release determination. And determination of batch quality can only be made after a thorough investigation is completed and determines to affect the batch quality. They're going to always tell you not to always use bad test results as an indication of product failure. You have to look at the big picture. It also depends on what the failure is. If it's a sterility failure, obviously we're going to have concernables. Why? Because out of how many thousands of samples that were on that line, how many thousands of examples, I'm going to use vaccines as an example. How many thousands of vaccines went through that fill line? And how many samples were pulled for sterility? The chances of you pulling 1 bad sample off of that fill line and have a bad sterility result are small. I mean, if you look at the statistics, it's very, very difficult for you to pull a bad sample off of a fill line. So when you do find one, it should be a big indication of batch product, sterility. And also, it would be concernable, especially if you're bulk passed, but now your individual samples are failing. What happened between the bulk container and my individual vaccine containers? Note again, caution should be taken in the case of inconclusive investigations or unconfirmed results. This is where I like to say I have to err on the side of caution. What risks are you willing to take when it comes to human lives? People are coming to you because they trust you. They come to you because they want the product that's going into them is to be safe. I don't like bringing COVID up in a presentation. I think it's getting so overrated and so overused. But now that we're going into the vaccine phase of COVID, people are questioning safety. People are questioning stability. They're questioning whether or not they want to take it because they feel it was manufactured quickly. It's our job as the manufacturers of these products to put faith in our clients, to show them that we have control over our processes and that we would never put a product on the market that we feel is unsafe for them to use. Prove them wrong, and they're never going to trust you again. Again, they come to us because they have no choice but to trust us. Are you willing to take that risk? And when you put it into that perspective, when you put that human element, that vaccine is going to go into my children. That vaccine is going to go into my family, my parents, my grandparents. I'm making it, but it's still going to impact my family. Would I want to put that product in my family? And if at the end of the day, as a quality person, I actually would question if I would allow my family to do this, I don't know if I would do that. I don't know if I would sell it if I wouldn't trust putting it even in my own body. So you have to almost put that human element into the aspects of these things. But realistically, folks, at the end of the day, err on the side of caution, because I don't want my clients to not trust anything I do moving forward. I want them to trust and have faith in me and my company and what it is my company stands for, okay, which is saving people, not hurting people. Common areas to look for and out of specification. Personnel qualification. Now, I'm always one to say I don't like using training or human error as an issue. I feel almost every human error that occurs can be a result of a quality system breakdown. So when it comes to human errors, yes, I'm a human, I have made mistakes. There are human errors involved, don't get me wrong. But can I build a quality system that prevents human errors from occurring in the first place? Because if you have good quality systems, you'll have fewer human errors. You have to build the system to remove the errors. But when it comes to people, inadequate training, and I'm not saying that they weren't trained, and I get frustrated when people bring up training because you're only as good as the person who trained you. And if the person who trained you wasn't the best trainer, you're a product of a poor trainer. Or you may be the product of someone who is very knowledgeable on how to run a test, but they're not a very good explainer. They're not a very good communicator. So if you can't train somebody else, then you shouldn't be a trainer. You might be an amazing analyst, but not everybody who can do a job can train on a job. Improper techniques. Yeah, we make mistakes, but I usually catch my mistakes and then bring that to my management's attention. In the middle of my test, I realize I'm using the wrong test method. I'm going to stop. I'm going to bring it to my manager's attention. We can invalidate and we can repeat. I would rather catch it up front than have an issue on the back end. Yes, it still might be an incident report because we're going to have another test being performed. But the reality is we caught the problem before it became a problem. Inexperienced reviewers and managers. Now, I don't want to call management out here, but do you understand the criticality of these tests? Do you understand what it means to be a reviewer? Do you understand how it means to manage other people? A lot of times in companies, we have technicians that get hired up into management positions, but we don't teach them how to be managers. We don't teach them how to manage their people and to be that quality oversight in our departments. So these are the kinds of skill trainings that we need to make sure our management come with. And also we have instrument competencies, differences between the sites and the contract labs as examples. Almost finished, folks. Again, unapproved suppliers or contract labs. Make sure you have an approval for supplier quality. Use of uncalibrated or damaged equipment. Unlabeled samples. Procedures were not clear that can lead to variability, and also missing data. Common areas to look for OOS, password sharing or system access if people can actually change data in the systems. Hybrid systems, if you're using paper and computer, no Part 11 audit trails. The systems are not backed up, so you can lose data, failure to retain raw data, failure to second verify original data, and again, altering or failing or changing failing data, which is technically fraud. So in summary, laboratory OOS management is under scrutiny, obviously. And you need to hold employees accountable to the quality standards you establish. Don't just train them and let them loose. If you do not hold them accountable or train them to superior quality standards, they're not going to meet your quality standards. A strong quality oversight is very, very important. Now, one of the things, again, that I have found as a concernable with COVID, and COVID has brought a lot of attention, as Hal was talking about. We need to start being more effective as a global company, as a global resource, is due to manufacturing and contact tracing and things like that, they're limiting how many people can be on an actual site in most cases. And I don't know how it is in India or elsewhere and globally, but in the US, they're very, very careful how many people are allowed on a manufacturing site to protect our manufacturing technicians. Quality oversight can be a big issue if we cannot be there to quality oversee our manufacturing processes. So we want to make sure that we're still holding employees accountable, even if we cannot be there 100% of the time. Employees used to see me on the shop floor all the time. They used to see me constantly walking up and down the floor. And because of that, they always were aware that quality was there and watching them. Well, that's not the case as much anymore. And because of that, there's a phrase that goes, when the cat's away, the mice will play. We don't want them to pick up bad habits. We don't want them to pick up bad techniques because people are not constantly watching them. But if you build that system, if you build that standard in from the beginning, that will not happen. But still, how do you plan to keep that quality oversight available when you can't be there? Just like our regulators are concerned, how am I supposed to audit a company and bring product into my country when I can't audit you because I can't travel to you? So we have to recreate, we have to reinvent the way we do things to make sure that we're still getting the same deliverables, even though we may or may not be able to be as present as we used to, okay? So these are the kinds of challenges that we are currently facing. And I feel for you guys, trust me, I do. This is not an easy job that we're up against right now. References, I'd like to show you guys some of the places I got some of my references from. Not every one of these references was directly mentioned in the presentation, but when it comes to global regulations, obviously FDA guidance for the industry, MHRA guidance for out-of-specification investigations. It's on the MHRA website. UDRELEX, 21 CFR, and there's actually PICS guidance out there actually for good practices for data management and integrity in regulated GMP, good documentation environments. Again, this is a draft document, but it's still a very, very good resource to have on hand. So I am going to go into questions at this point, and I know in case we run out of time, it was an honor and a privilege to be part of this conference. I apologize for some of my technical issues. Again, I'm not a lab person. I'm not a computer person. I'm actually a hands-on lab person. So it just goes to show you that sometimes even we have to reinvent ourselves when it comes to these webinars and being able to communicate with folks globally, even though we cannot be with you.